Let's get started. Welcome everybody to today's talk by Tracy Kivell from the Max Planck Institute in Leipzig. And I just wanted to briefly introduce Tracy to you. Tracy did her PhD in Toronto and was then for a couple of years an assistant professor at Duke, if I'm right.
Yep.
Before moving as a postdoc to the Max Planck Institute for Evolutionary Anthropology in Leipzig, the Department of Human Evolution at that time. And where I think during your first presentation, I learned a lot about knuckle walking.
Yes,
anyway,
my terrible interview, but yes.
Yes. So Tracy spent then a while in Leipzig, and before she moved as I think first senior lecturer and then professor to Kent, the University of Kent in the UK, where she stayed until 2023, sorry, 10 years, I think.
Yes.
And when she moved back to Leipzig as the new director of the Department of Human Origins now. And so we are very happy to have you here today and looking forward to your talk on the evolution of the human hand. Thanks.
Great. Thank you, Philip. Thank you. And thank you for the invitation. And thank you all for coming. And guten Morgen. Okay, so spacebar. Okay, spacebar. Before I forget, is that working? No, hold on. There we go. Okay, so as did that work, Maeve? Yeah. Okay, great. So as Philip mentioned, I'm going to talk about the evolution of the human hand. This has been my main area of interest since my PhD, but this is, although it's my main focus, I'm going to talk about work that is presented by a large team of people, and so I'm going to try and on each slide sort of highlight who's been doing what, because as many of the more senior people might realize, the more senior you get, the less research you actually do, actually know how to do. So it's much smarter people than me that are actually doing the work. But generally, my research is driven by trying to understand the evolution of the human hand, and why and how and when we might have evolved from a hand that looked something like a gorilla or a chimpanzee that is used for locomotion and for other manipulative behaviors like grooming and food manipulation and occasionally tool use, to our amazing hands that typify humans, that have this really quite remarkable dexterity, are capable of really precise and forceful manipulation and really complex tool use. And we studied this through, of course, looking at the fossil record and what those bones of our fossil hominin ancestors might tell us about how they use their hands. But we also do this through the study of internal bone structure, which I'll talk about today. And then in order to sort of understand those two things, we need to also better understand how living apes today use their hands in the wild. And good morning, Karen. And also through experimental biomechanics. So I'll try and touch on all of these topics a little bit this morning. And I'm interested in the hand because it helps us to address two, fundamental questions about human evolution. One being, when did our fossil hominin relatives sort of stop using their hands for locomotion and climbing and potentially suspending in trees? And also, when did we sort of transition from more maybe simple and occasional tool use to more complex and habitual and obligatory tool use? And of course, the archaeological record tells us a lot about that as well. But how do we see these signals in our hand morphology? And traditionally, the idea has been that if you are a hominin that is still using your hands regularly for climbing in the trees and for locomotion, that you're not going to be able to be really good at also using complex tools habitually, so that these two things are mutually exclusive. And so just to, I think most people in the room are familiar with some of these characters, but just to make sure we're all on the same page. So this is just a spattering of some of the fossil hominins that we have over the last 7 million years. And today I'm just going to focus on two of those, one being Australopithecus sediba and the other being Homo naledi. And so sediba is around 2 million years old, found in South Africa, and falls at this transitional time period in our evolutionary history in terms of body plan. So she still has, obviously is a biped and is a hominin, but still has longer arms, like an ape, still has a small brain. And found at a time that is sort of at this transition between sort of what I'm going to call simple stone tool use. I apologize for the gross oversimplification of your archaeological record here. And then this sort of transition into sort of more complex Acheulean hand axes, for example. And then Homo naledi, which is much later or more recent in time, still has a body plan that is relatively similar to sediba in terms of having longer arms, a small brain, but is found around 300,000 years ago, and which is at a time that is obviously contemporaneous with Neanderthals, Homo erectus, as well as the first evidence of Homo sapiens. So I'm just going to focus on these two. And obviously, it's also at a time when we have sort of committed tool use. Again, very gross oversimplification. And for someone who is interested in hands, these two fossils were really quite remarkable for me. So Lee Berger is the discoverer of both of these amazing fossil finds in South Africa in the cradle of humankind. And both of these skeletons here came with associated hand skeletons. So these skeletons were found in semi-articulation. In the case of sediba, it went along with a beautifully preserved complete upper limb, which is really rare in the early hominin fossil record. And in the case of naledi, this is one individual's hand, but there are multiple, upwards of 20 individuals found in this rising star cave system. So we don't know sort of what arm and body it went with, but we know that those bones go together because they were found together. Okay, so when these were first discovered, we sort of looked at the external morphology and said, let's try and, what was sediba and naledi doing with their hands in terms of locomotion, and what were they doing in terms of tool use? Did they show signals of sort of more simple, occasional tool use, or more potentially habitual and complex tool use? And to do this, so we focused first on the external morphology and compared it to humans and chimpanzees and other fossil hominins. And there are many things that are different between a human and a chimpanzee hand that relate to or are thought to relate to our sort of very precise and amazing dexterity. I'll just focus on a couple of these key features. One being the relative length of our thumbs relative to the length of the fingers. So humans have really long thumbs that are big and powerful with extra muscles. And humans have relatively short thumbs and long and curved finger bones, which would be useful for climbing in trees. And yes, so the curvature of those fingers in a chimp are facilitate grasping during locomotion, and our own fingers are relatively straight. And so when we looked at the sediba and naledi hands, they have basically a hand skeleton that looks more human-like in terms of the overall proportions. So they have relatively long thumb and short fingers. In the case of sediba, it has an even longer thumb than what we find in humans today, which was really surprising. And naledi also has this long thumb and a really powerful, like one that looks like it's quite powerful. And it also has wrist bones that look more like humans and Neanderthals, which suggested to us that, but it has curved fingers. Okay. So it has all these sort of human-like features, but the finger bones are quite curved. They're a little bit curved in sediba. In the naledi, they're remarkably curved, both the proximal ones and the intermediate ones. So it's this weird mix of morphology. And so we said, based on just the external shape of the bones, that Sediba showed morphology that would suggest, yes, it was still using its hands for locomotion, but also showed, having this long thumb, for example, would facilitate bringing our thumb to our fingers and would facilitate manipulation and potential tool use. And with Naledi, the same kind of story, but because it had wrist bones that looked like Neanderthals and humans, we said, well, actually it looks like more complex kind of tool use here, that it was more habitual. Okay, so that's what we could say about the capabilities, the potential behavior from the external shape of the bones. But what I'm really interested in is, what are they actually doing? So obviously behavior can evolve faster than morphology. And so we wanted to know, well, what are these actual individuals doing with their hands? And we think the key to addressing that question is really looking at the internal bone structure and applying this sort of long, sort of well-established concept of bone functional adaptation. So our bones are made up, just to make sure we're all on the same page. We have cortical bone, that hard outside shell, and then we have trabecular bone, which is the meshy network of bone that's found underneath our joint surfaces. And because bone is a living tissue, it is capable of modeling and remodeling throughout life, depending on how we use our skeleton. So where the loads are higher, more bone will be added, and where load or stress is lower, bone can be removed. And it does this process, it does this throughout our lifetime, which means that it can provide a more clear functional signal of how you actually used or loaded that bone during your lifetime. And a perfect example of this is if you think of the right and left arms of Serena Williams. Her dominant right arm is much bigger than her left. And if we were to CT scan her arms, you would see that she has a lot more cortical bone in the humerus of her dominant arm. So this is an example of this concept of bone functional adaptation, reflecting her individual behavior. And so we do this through actually colleagues here in Vienna at the Technical University of Vienna. And we apply sort of holistic methods to quantify the entire trabecular structure within a given bone. So here we're just looking at the third metacarpal, the bone in the middle of your palm. And there are many complex steps here, of which I now don't know how to do it anymore, but people in the group do. And the important part here is just, I'm going to show you a bunch of color maps in this talk today. And just recognizing that where you see red is where there's lots of bone, and where you see blue, there's a lot less bone. And we make the underlying assumption that where there's red, that's where loading is higher, and where it's blue, that's where loading is lower. And we use that to try and reconstruct how a particular joint or bone was used during life. And we've shown previously, this is the work of Zodi Sagai during her PhD, also at the Max Planck at the time, that when we look at living apes, that their structure of their metacarpals reflects how they use their hands during locomotion. So in a chimpanzee that spends most of its time knuckle walking, this is looking at the head of the metacarpal here, that it's in this sort of hyperextended position, and the most red part of the head of the metacarpal is in this sort of reflects this kind of dorsal positioning of the phalanges on the metacarpal head. So it reflects this knuckle walking posture. In contrast, if we look at an orangutan who spends most of its time grasping branches with flexed fingers, in which it's going to load its metacarpal more on a palmar surface of its here, that the red bit is more on this side. So we see these sort of correlations in how living apes use their hands during locomotion, and the differences in their trabecular structure. And Chris Dunmore, for his PhD, he looked at, so Zodi just looked at one of these metacarpal bones, and he looked across all four of them, and saw that what was unusual about humans is that we see sort of variation across the hand in how the sort of these color maps are divided. So we see lots of red this is the second to the fifth metacarpal. And when we use our hands during daily activities, we most often use our second and third digits. And this is reflected in this sort of diverse pattern across our palm that reflects how we sort of use our hands in daily life. So we can apply these sort of patterns that we see in living apes and humans to the fossil record. And so Chris looked at the trabecular structure of the Sediba metacarpals. And so this is what Sediba looks like, again, from the second to the fifth side, or the pinky finger side of the hand. And in comparison to living apes, humans, and some other fossil hominins. And Sediba falls out next to orangutans. So basically showing that it has a trabecular structure that looks more like it's loading its fingers and its palm in a way that orangutans do. So using more sort of what we call a power grasp, where you use all of your digits. And maybe that's for climbing in trees, maybe that's for grasping big tools. But this is what at least the trabecular bone structure tells us about how it was the position of the fingers during its habitual daily activities, or at least that's what we assume. Okay. And this conclusion about Sediba having sort of an ape-like fingers is supported by my PhD, Summer Zaida, who's just finished. She looked at the distribution of cortical bone in the fingers, so in the proximal and the intermediate phalanges, across apes and other fossil hominins. And so this is looking at the hard outer shell just among the shaft. Again, where we see red, it means thick cortex, where we see blue, it's much thinner. And in chimpanzees, orangutans, as well as gorillas, you can see that the red bits are on these regions here on the palmar surface, which are what we call flexor sheath ridges. So this is where soft tissues attach that reflect using your fingers powerfully in sort of flexed postures. And in humans, show a very distinct pattern in which the biggest or the most thick cortical bone is on the dorsal part of the phalanges, on the backside of the fingers, which we interpret as being sort of, loading during manipulation, but of fingers that are relatively straight. Chimpanzees and orangutans have these curved fingers, which also helps to distribute sort of these large loads that they incur during locomotion. So she looked at Sediba, and Sediba shows a pattern that looks much more like the apes and not like humans. So again, it fits this sort of, that matches the pattern that we saw with the metacarpals, in terms of it showing a more ape-like pattern in the fingers and the palm, and not this manipulation signal we see in humans. Okay, so ape-like signal, there you go. Okay, so that's the fingers, but what about the thumb? So I talked about how our big, long, powerful thumbs are really important for our dexterity, and in comparison to the tiny thumbs that chimps and gorillas have, which are so short they don't even load them during knuckle walking, and were traditionally thought to be actually so small that they're relatively useless, that they're so small they can't even use them for grasping during locomotion, versus humans that have these really powerful ones that are, very important for tool use and our daily activities. So we also looked at the trabecular structure of the thumb. This is just at the first metacarpal, so this bone at the base of your thumb. And what I'm showing you here is we've taken that first metacarpal and cut it in half. So you can sort of see the section. I've tried to depict that in the top corner there. And we're looking at, our cross section to that first metacarpal and the trabecular bone that's inside. And we can see that there's some quite distinct differences between a chimp and a modern human. So chimps have lots of red. They have lots of trabecular bone in their first metacarpal. And if we just focus on the base here, this is where it would articulate with your wrist. There's lots of red. It's all over the place. Whereas in humans, there's far less. There's a lot less trabecular bone, and it's all concentrated to one side, to this palmar side, which is the position how we load that joint when we oppose our thumb towards our fingers. So we also looked at Neanderthals, and they show a very similar pattern to what we see in humans. If we look at Australopithecus africanus from 3 to 2 million years old in South Africa, it shows sort of a hybrid pattern, so it's got more trabecular bone in the shaft, but it is also concentrated on this one side that is more human-like. And if we look at Sediba and Naledi, they show a pattern that is more similar to humans. So less trabecular bone, and again, it's all concentrated to this one side, which is like loading the thumb towards the fingers in sort of, yeah, forceful opposition to your, or forceful manipulation. Okay, and so this is that same principal component analysis that I showed before. So I should say we take all those color map data, throw it into a principal component. And here's when Chris looked at the fingers, or at the metacarpal, of the palm, and showed that it looked like an orangutan. And if he also looked at the thumb, and Sediba actually falls within the human range of variation in its thumb. So there's this sort of dual signal, one for the fingers that's more ape-like, and one for the thumb that is more human-like. Okay, so with that, we sort of said, okay, does the trabecular structure actually seems to support that initial our initial conclusions just based on the shape of the bones, that we see an ape-like signal in the fingers suggesting locomotion, but a more human-like sort of loading or signal in the thumb. And the same with Naledi, that we have these, especially with these curved fingers, that there's very strong signal for locomotion, but that there's potentially signals that human-like loading of the thumb. I say human-like loading, and that's as far as I try to go. Like now we use this during tool use. We use this and we can show modern humans with our modern human hands trying to make stone tools in a hominin-like way that this is how they do it. And of course, we don't know how they would have grasped a tool or what size those tools were. And certainly if we could actually find tools that were associated directly with Sediba or Naledi, that would be really helpful. But at least the trabecular structure sort of fit with how we interpreted the external shape. Okay, and in the case here, where I talked about how we used to think that these two things were mutually exclusive. So if you were using your hands for locomotion, you couldn't be a really good tool user. And I think in the case of Naledi, that in particular, that's actually not the case. That in Naledi, that we see both of these things happening in the same hand. That there's adaptations for both of these functions. Okay, but so can we be confident about these functional interpretations? So at the end of the day, although we quantify this trabecular bone and we can do this statistically and feel, we can match it to the way that we can see apes use their hands and what we know about their locomotion and tool use in the wild, there's a lot we don't know about how they actually use their hands and how they load their hands. And so the next step here, I think, requires this integrative approach of biomechanics and understanding how apes, what they do in the wild. Okay, so I mentioned that, chimps have these tiny little thumbs that we used to think, are relatively useless in terms of they're just too small to be sort of functional in some way. But that was based on the fact that no one had really looked at how chimps or gorillas use their hands when they're climbing in the wild. And so Johanna Neufus, this was her PhD project where she went to the Bwindi in Uganda and looked at mountain gorillas as well as sanctuary chimps living in Chimfunji in Zambia, and looked at how they used their hands when they were climbing and suspending, and showed actually the thumb is used all the time. That might seem like a really obvious thing. Like, of course the thumb is, of course it's functional for something, or otherwise I don't. But it hadn't been. No one had actually studied it before, and she quantified how they use it and what postures they use it, and that it's actually seems to be really important. And here's just because I like to show, videos are more interesting. This is not great video for showing how a gorilla uses its hands. I took this myself. I had pleasure of going to Burundi in Uganda in November. And so I'm showing this gorilla climbing down a tree, in part because I think it's really cool, and two, because this is a silverback up in a tree. And I think we often think of gorillas, and I've done this myself, as terrestrial, largely terrestrial knuckle walking African apes. And even the silverbacks are spending, at least at Burundi, a lot of time in the trees. And so this is something I think we need to think more about when we interpret their skeletal morphology. Gorillas will also use their hands for manipulation. This I also took in Burundi. I quite like this guy because he's clearly broken his third digit, and is showing some quite, he's eating his breakfast and showing, manipulating this plant while he's eating. It also, as you can see, he's broken his third digit and he's telling me what he thinks of me interrupting his breakfast. I think he wasn't so pleased I was standing there. And this was another, aside of like, that finger has clearly been broken for a very long time, and he knuckle walks with it sort of stuck out like this, and it seems to be perfectly, thanks Steph, happy manipulating and doing whatever and climbing with this broken finger with no problems at all. Okay, so Johanna also looked at how gorillas and chimps use their hands during manipulation, and showed that, and so have others. This Byrne et al. Had done this in 2001 at the Karasoki gorillas, but showed that they are really capable of quite amazing dexterity, using their hands, each hand in different ways. And Johanna showed actually the way that they process some of these plants, especially when they're holding on and pulling them with a plant with their teeth, that they're probably loading their thumbs actually really quite forcefully, which is something that we think only humans can do. And then she also looked at how bonobos nutcrack. So these are bonobos at the sanctuary of Lola. Bonobo. And although we know bonobos don't often use tools in the wild, they are, these sanctuary ones know how to nutcrack, and they're actually very good at it, and they do it just as efficiently as chimpanzees do in the wild. And they use their thumbs when they're doing it with these really awkward big hammerstones. And so I think it just sort of drove home that actually that thumb is going to be really important during locomotion, as well as various manipulative activities. And so, maybe that explains why they've got this whole bunch of trabecular bone in this thumb that we sort of typically thought was relatively useless. What time am I at? Okay. Okay, so that was nature. So and I think looking at behavior in a natural environment is really, really important, and we haven't done enough of it, but there are limitations and a lot of challenges to collecting that kind of data. And there are things that we can do in the zoo that we can't do in the wild. And so one of these was to sort of understand, well, if they're using their thumbs for locomotion, what load or stress or pressure is that thumb actually experiencing when they're climbing up a tree or suspending? And so this was work done by my postdoc, Diana Samuel, as well as colleagues at the Plankendale Zoo in Belgium. And it was the captive bonobos, in which we wrapped a pressure mat. So that's highlighted in yellow there where the white bit is. That's a pressure mat on this pole that we put both vertically. You can see the sort of chaos of bonobos in the zoo. And then we put it horizontally so we could also get suspension, as well as arboreal knuckle walking on top of the branch. And then Diana was very patient and just waited. We weren't allowed to provision them or sort of encourage them to climb up this pole. So she just had to wait. It took 9 months of data collection to get decent sample sizes. But in the end, we collected, so we could have video data. And so anytime they touched the pressure mat, the cameras would go off and the pressure mat would start recording. And we can see that the thumb is being used to grasp the pole every time they climb up it. But when we looked at the pressure data, actually, the thumb, so we quantified how much pressure is being experienced by the palm, the fingers, and the thumb. And the thumb is experiencing really quite a lot less pressure. And it's only out of the 37 different trials, only 12 of them actually loaded the thumb. In the other ones, there was no pressure registered on the pressure mat at all. And this was the similar case for suspension. So again, we can see from the camera that the thumb is touching and is grasping the substrate. But when you look at the pressure data, there's no pressure being registered. There's no load being experienced by that thumb. Or there's some, but it's not high enough to be registered by our pressure mat. And so this was a bit surprising to us because it thought, we can see from nature that they're using it, but it suggests that actually there's not a lot of stress being experienced by that thumb during this locomotion. So it leaves us still with the question of why is there a whole bunch of trabecular bone in there? Because we would expect there to be relatively little if it's not being loaded very much. And I have many other ideas about why that is that we can talk about later if you like. There's a major caveat here in that in the zoo, we can only look at one size of a substrate. We're looking at captive, not wild individuals. And the zoo environment is very sort of structured and doesn't offer the same sort of complexity and versatility that they need to have in a wild environment. But it was useful data, and it's data we can't collect in the wild. Okay, we also did the same for humans, and humans are way more cooperative than bonobos, and you can tell them what to do. So we use that same pressure mat. This is a master's project of Victoria Lockwood, in which we asked humans, including myself, to suspend and climb on a pole, and also measured the pressure. And what we found, which I forgot to highlight with the bonobos, is that actually when they're climbing, most of the pressure is being experienced by the fingers, and particularly in the middle of the hand. So we just say the third digit for simplicity, and relatively little for the thumb. So humans show the same pattern when we are climbing or suspending. And so this is sort of goes back to these two big questions. So one, this was sort of biomechanical data that we could use to address this question of locomotion. But what about tool use? And so we did Erin-Marie Williams at Tala, along with Alistair Key, collected data using, again, with pressure, but using a pressure glove this time on humans, and in which we had sensors across all the thumb and all the fingers, and asked them to do a variety of different tool making and tool use activities. So we had them do some percussive activities that we think fossil hominins might have done, like getting marrow, napping, nut cracking, as well as butchering with hand axes and flakes of various sizes. And we measured the pressure experienced by their digits when they did these activities. And here I'm just showing, again, this is a heat map of the different pressures. So where you see red, it's higher pressure. Where you see purple, it's low. And first of all, like across, and then these are all the different activities along the x-axis. And you can see that, first of all, there's lots of purple for nut cracking. So not a lot of load or pressure is experienced by the digits during nut cracking, which might sort of inform us about chimpanzee behavior and hand morphology. And that the highest pressures are incurred during marrow acquisition and napping, which is essentially the same kind of behavior. You're just banging two things together. And that almost for all of these activities, the highest pressure is being experienced by the thumb. The only exception was you cutting with a small flake, in which your index finger experiences a little bit more pressure. But there's an overall pattern here that during tool activities, the thumb is experiencing the most load across the hand. And so if we summarize all of this, so we look at locomotor activities. In bonobos, during all of these different locomotor activities, the pressure data tells us that it's the third digit or the fingers that are experiencing the highest amount of load. Same within humans, and that the thumb doesn't experience very much. But during tool use behaviors, it's the opposite. The thumb is experiencing the most pressure, and the fingers relatively less. Unfortunately, collecting this data for apes in terms of tool use is very challenging, if not impossible. But for now, we just sort of think, okay, if we look back, if we're trying to sort of figure out, really tease apart the functional signals in the fossil record, that the fingers maybe will tell us a little bit more about locomotion, and the thumb will tell us a little bit more about manipulation. Okay, so then to summarize, so I think here we find that, again, the experimental data then adds this next level of evidence that helps us feel a little bit more confident about these initial interpretations of the fossil morphology. And so, yes, can we feel confident? Yes, I do feel slightly more confident, but there's still many unanswered questions. And for me, I thought, well, if the thumb is not actually that important during locomotion, it may have been, in an evolutionary sense, sort of more free to adapt to the needs of manipulation. If we assume a Sediba or Naledi loads its hands in the same way a human and a bonobo does when it's climbing or suspending. And that doesn't mean that they weren't using their fingers for tool use. It means that likely, given that the pressure or the load that your fingers experience during locomotion is much higher than they are when you're doing manipulation, that the locomotion is going to override any kind of manipulation signal in the fingers. They weren't using tools just with their thumbs to get that point across. And so, yes, this sort of overall idea that Sediba and Naledi were using their hands both for locomotion and for tool use is one that I think is now borne out with a number of different lines of evidence, and that here we have these sort of dual functional signals, and I like to think of it being a sort of hand morphology that's adapted to two biological roles, that it's not necessarily a compromise between the two, but that our hand is capable of doing both of these things in these hominins. Okay, and so I feel like I often talk about Sediba and Naledi, and it gets really boring in my own head. And that's partly because they are still the most beautiful and most complete early hominin hand fossils. Naledi is not early, it's relatively recent, but I think of it more as an early hominin in the sense of its morphology is quite early hominin-like. But we are also working on the 087 hand of Homo habilis, as well as this composite hand of Homo floresiensis. And they show different signals to Naledi and Sediba. So in each case, there's sort of these different mosaics of functional signals within the fingers and the thumbs, or at least what's preserved. And so I think all of these amazing creatures were finding slightly different ways of dealing with these sort of dual functional needs of their hands. Okay, my final two bits is whenever I talk about functional morphology, I think it's important to talk about the GOAT principle, which Mark Meyer, at least I think coined this sort of term in 2016. I apologize if someone else has done it before, but it's the fact that we were using bone to reconstruct behavior in the past, because that's, for the most part, all that is preserved in the fossil record most of the time. But, if we were to find a fossil mountain goat and reconstruct its behavior from its skeleton, we would never probably think that they climbed in trees or were on these precarious rock faces. And so there's always that very large caveat of what we can actually extract, and the assumptions we're making that the bone is reflecting habitual behavior during life, but whether we can capture all the things that we're doing is very unlikely. There's also the very large caveat that bone is reflecting not just function and how we load our skeleton, but many, many other things, including like it changes throughout ontogeny. While our bone is growing, it's more able to reflect these behaviors than it is later in life. Hormones, genetics, pregnancy, the differences between age and sexes, and even the microbiome affects your trabecular bone. So there's lots of different factors that that play into the way that our skeletons look, and we don't really have a full understanding of how those work even in humans, let alone how they might have worked in fossil hominins. And so part of my new department is going to try and address some of those questions to try and get at what we can feel confident about in terms of function. So we're going to find hopefully a lot of willing participants. Any of you want to come to Leipzig that do unusual and repetitive and wonderful things with their hands and feet, like play instruments, carpenters, gymnasts, marathon runners, mountain climbers, rock climbers, people who do things that are unusual with their skeletons and have done them for a long time, and that we can scan their hands and feet and see if we can see evidence of this in their skeletons. This also includes work that I'm doing with these lovely fellows that we're going to try and actually also collect data on the internal bone structure of hunter-gatherers that engage in behaviors that are more relevant to understanding the human fossil record than us typing on computers and stuff like this. And so in particular, the Hadza in Tanzania and the Batek in Malaysia that still use stone and wood tools, and also use and climb trees, often unassisted. And so I think there's it's these kinds of samples that'll help us better understand what we can and cannot say from the fossil record. Okay, and so with that, I just want to thank funding and all the museums that made all their specimens available, because I can't do that without their help, and the many, many collaborators that there's more than this. These are the phases I have so far that make this all happen and do most of the hard work, and I just stand up here and claim it as my own. So and thank you all for listening.